Today on Standing on the Shoulders of Giants, we have with us Amanda Vondras, who is a molecular biologist, which is pretty hardcore. I initially thought she was a geneticist, but have since corrected myself, thank God. But you do work a lot with genetics. You've done some really interesting things with grapes and wine and things of this nature in particular, which I would love to get into. But first, let's hear a little bit about Amanda herself. You have a pretty strong background, Cornell, Oregon State, PhD, etc. Hit me. What's the background look like?
So when I was an undergraduate student at Cornell, I became obsessed with grapes. I love everything about them. I love the beauty of where they're grown. I love smells, so like aroma of not just wine, but also perfumes and beer, and I can nerd out on smells like all day. My first mentor was actually a chemist, not a biologist, and I had taken his class on wine chemistry and composition because I was enthusiastic about like organic chemistry. I was just like, oh, wine, that's cool. Like, what's this about? So I took his class, became enamored with the subject, and I asked if I could be his minion, like in your lab, like I'll wash dishes, like please, can I get involved in research? Also, if you're an undergraduate student at a university that does research, like that's a great way to figure out, do I want to be a scientist? What do I want to do? Is just kind of like connect with a subject, get involved in research in a subject that you're interested in. But he was like, I'll do you one better. Like there's money available that you can apply for. You can write a grant, get money as an undergraduate student to do your own research project. So he helped me, apply for the grant. I got money as an undergrad, which is not super common, but was great because I could do my own research freely as an undergrad, and I got my own little lab space, and that's how I became sure, I want to be a scientist. I'd always kind of wanted to be, but now that I had put myself in that environment, I was certain, like, this is what I should do. And if you want to be a scientist, much of the time you have to have a PhD. So I was like, where should I get my PhD? And he recommended, like, find a project that you're really interested in. And he also encouraged me to go to this one lab at Oregon State. So that's why I moved to Oregon.
It's also gorgeous.
And it's also gorgeous. So like that's the other piece of, working in grape is that, every summer and fall you're working mostly just like designing your experiments, you're, sampling and tying strings on individual berries for, to collect them later. It's the most beautiful place to work. I've had a variety of jobs growing up, but like I've never worked anywhere else that like regularly fills me with awe. And that's kind of why I stuck in grapes for about a decade. And then after my PhD, I went to UC Davis. So I had been presenting. If you're a grad student, you make posters and you present them at conferences, and then you stand by the poster and people walk by, and if they're interested, you tell them, you try and give them like a few sentences, this is what I do, and if they're interested, they talk longer. Anyway, so I met who would be my postdoc advisor at a conference. He saw my poster and was like, when you finish your PhD, you will come work for me. And I was like, okay. And
aggressive, I like it.
Yeah, it was great. And then every once in a while, he'd like email me like, are you done yet? That kind of deal. So I went to go work for him at UC Davis, and I worked for him for 5 years. I'm sure we'll talk about some of the work I did there.
Yeah.
And UC Davis is like Grapetopia to do grape research. I worked at the Robert Mondavi Institute there. The labs are these beautiful glass buildings, and in the courtyard there's olive trees and pomegranate bushes. Are they bushes? I don't know.
Yeah.
Pomegranate plants. There's this like beautiful garden, and you just get to see that every day. And then there's a dedicated experimental vineyard close by specifically for studying my virus of interest, grape leaf roll virus, grapevine leaf roll virus. So that's kind of my background. And then you realize, oh crap, I know a lot about grapes. Like, I can't be a postdoc forever. What am I going to do? I don't want to be an academic. I started looking for a job, and I got recruited by UNC Comprehensive Cancer Center. So I like really switched gears and I became a bioinformatician at the UNC Lineberger Cancer Center. And I did that for about a year. And now I'm like totally out of academia and I'm working at a startup.
Nice. A bunch of different stuff I want to unpack.
Yeah, sorry.
There. I mean, no, that's great.
That's the full.
That's amazing. There's a bunch of
lineage.
different stuff I want to unpack because wow, you've had a pretty great career.
Thank you.
Couple of different things inside of there. First of all, just a comment. Mondavi, specifically their cabs, like a nice 97 Reserve. Oh my word, is that
Have you been to
unbelievable? I've been to, yes. So I lived in Northern California for a while. I've been all over Napa, the Camus wineries, the Silver Oak. That's probably my favorite. I've been to Opus. I've been to Mondavi. Been to a few different spots. But yeah, amazing that they're doing that. And what a great treat in Oregon to have done that. First thing I want to unpack though is, if one wanted to get into molecular biology, how would one do that? And for that matter, why? Because not to draw the question out, but it seems like almost everything, like literally almost everything that's alive, is effectively downstream of molecular biology, right? I mean, it's the root of everything. So why was that so interesting for you? That's such a great subject, and not a lot of people necessarily even know, like maybe not even what you do or like how you get into that. So can we chat about that for a sec?
Sure. So yeah, my primary interest, or the bottom level of my interests are genome structure, function, and organization. Most of the time as it relates to grapes, but then most recently as it relates to cancer. And if you're interested in this kind of thing, if you're a young student, that's usually I would guess that's the path of least resistance. If you're interested in molecular biology, like find a lab or a professor whose work you're interested in, like on any university page, you can click through like professors, what they do, and just be like, prior to grape, I actually also worked a little bit in a tomato lab. And I was kind of a grunt, So just like say, I'm interested in this, get your foot in the door. There will probably be a lot of dishwashing, but you'll learn a lot of useful skills that you can then parlay into doing your own research and competently. If you're not a student though, like if you're a hobbyist, let's say, and you just want to get into this, I know there is a community of biohackers in Austin where we are now. One of them who is like super cool and actually runs a company to facilitate DIY biohacking kind of stuff. His name is Joe Zahner. And right. Joe Zahner runs a company called the Odin. And the Odin sells basically like kits for doing molecular biology in your garage, things like that. So if you want to like fool around with molecular biology, like that's another way to do that.
And I think it's the-odin.com, correct? Something of this nature.
I'm sorry, I don't know.
I think it is. If it's not, it's Joe Zahner, J-O, and then Z-A-Y-N-E-R. I think that's correct.
Yep.
I visited that site. It was pretty interesting stuff. So that's the how. Now I want to go into the more of the why. Why does why does someone know that they're interested in this in the first place? Like, are there specific signals? You're just like, oh, I'm really interested in like super tiny things.
Oh, well, okay. So the grape stuff I think is pretty obvious. Like even if you're not a molecular biologist, I think the average person can understand like why you would appreciate the beauty of vineyards and wine and like its connection to human history and all of this stuff. That's the easy thing. And then maybe it's obvious, maybe it's not obvious. I think humans in general are obsessed with things that they can't see. And you can't see things with the naked eye, molecular level. So that's my, how do I say this? That interest in me manifests as like, oh, I want to be a molecular biologist. Like, I want to understand that which is unseeable, like what's going on behind the scenes, like what makes me, what differentiates you from me. I think
Right,
that's
we've
really
got
cool.
these like clumps of cells that are you've got your clump of cells over here, I got my clump of cells over here, and you're going, cool. I get that.
I'm also interested in there's kind of universals. So like obviously there's like things that differentiate us from elephants and things like that, but the underlying building blocks of life are universal, and that is really, really cool. So you have this DNA, it's still DNA, like in the elephant and in me, it's just different.
It's that
And
CTGA or whatever.
ATCG.
ATCG, sorry.
And I just think that is so cool that you have these building blocks of life, and if you assemble them in this particular way, you get a, and if you assemble them in this particular way, you get a, and then understand the relationship between that particular sequences and certain functions. So I mentioned before, my first toe into grape world was like, oh, I'm interested in chemistry, actually, and aroma. Well, there's a molecular, biological, genetic basis for aroma. So that I'm interested in the connection between what's at the bottom, like the bottom layer, biologically speaking, and the output, things that I enjoy or can affect my life.
Yeah, that's big. No, seriously, that's big. I definitely appreciate that, because I had something reasonably similar in the computer world where I was just like, I don't want to run programs. I want to program. Like I want to build things that people use, and then I want to understand, wait, how does this program translate into machine code, and then how does that translate effectively into electrical engineering in the chip, and so on and so forth. So I can definitely appreciate where you were coming from. I like that.
Oh, thank you.
So if I break it all the way down, and correct me here, but like it just seems like it's more of like you have an intense curiosity about life.
Yes. Yeah, that's a much better way of putting it, actually. It's more succinct.
Well, I mean, but your way was much more descriptive.
I have that's a great way of putting it. I'm not going to be able to put it better than that.
Excellent. Well, okay, so clearly now, and again, I'm going to go right back to the beginning statement, because there was a lot to unpack. There was wine, there was cancer, and then there's your startup. There's a lot in there. How about we hit up wine first?
All right.
So this is a good thing. I'm a big fan. I'm a red guy, although I do enjoy some champagnes.
Champagne is awesome.
Yeah, I love champagnes. But basically, when we talk about molecular biology, at least from my layman's understanding, it's a grape is a grape, and we can kind of all agree to for some definition of this is a grape.
You can squish it and eat it.
Right, but they're very different grapes, like wildly different grapes, and sometimes grapes are so close together. I mean, we sort of spoke a little bit offline about this, but sometimes grapes are so close together that it's like actually that's the same grape. It just has a weird genetic variation. Can we talk through a little bit of that? I can't. What was it? Was it Pinot? I can't remember.
Yeah. So
that's right.
So there are different cultivars. There are also clones that happen to have different names. So, for example, Pinot Noir, Pinot Gris, Pinot Blanc, and Pinot Meunier are all clones of one another. That is that they have somatic mutations. So there's no sex going on. So the variability between them is not caused by mating, but there are mutations that just occur over time that account for consequential differences between them. So Pinot Noir is red, Pinot Gris is gray, Pinot Blanc is white. So that's just somatic variation. And then between different grape varieties, so for example, Cabernet Sauvignon is
my favorite.
Yes, your favorite is the offspring of Sauvignon Blanc and Cabernet Franc. I hope I said that right. And this is just brief little aside. What's really cool about grape is that when you actually look at the genome, there's a lot of what's called structural variation. So you think most of the time of a diploid organism, that is, for every gene you get one copy from mom and one copy from dad.
Okay.
In grape, the crosses are relatively distant, which means that in some cases you only get one copy from one of the parents. So you get what's called hemizygosity. And just someone who studies grape, as a consequence, you can't use just like one. There's one reference grape genome that's been used for most of the studies done, but there's a lot of interest in sequencing and assembling a lot of different grape genomes so that you can fully represent all of the variable gene content that can occur between different cultivars.
You mentioned two different words there, and I need some help here. You mentioned, I think, structural and somatic?
Yeah.
What's the difference between those two?
Oh, so a structural variation is just, an example would be, let's say you have a reference genome, so like some representative sequence.
Yes.
You can have individual positions in the genome that are different, called SNPs, or single nucleotide polymorphisms. You can have indels, and these are small insertions or deletions, let's say under 100 base pairs, or 100 A, T, Cs, or Gs, no more than that. So either an insertion that size or a removal that size. You can also have, so structural variations can be really big inversions where relative to the reference, the sample you're looking at, an entire region has been turned around. You can have duplications, large deletions. What's another one? Large insertions, large deletions, large inversions. You can also have transpositions. So this sequence is over here in the reference. Oh, and it moved way over here in this other, in your sample of interest.
Does that matter? I'm asking because does that matter in DNA? Because is it read in a particular fashion, like the order matters?
Oh, yes. Asterisk. So if you're talking about, does structural variation matter? Yes. Does the order matter? It can. So for example, with some really large insertions or deletions, you could be inserting or deleting gene-containing blocks. And if that gene plays some role in the characteristic of a particular wine or something like that, if that the presence or absence of that gene could be consequential. In terms of order, so I might deviate slightly from the topic of structural variation. So just why does order matter? So if you look at a gene in the genome, it is made of, well, let's see. I don't know if I can like directly talk about this in terms of structural variation, but let me try. So if you can move around the entirety of the gene plus the stuff that regulates it, maybe it wouldn't be consequential, but I'm not certain.
Okay.
So so genes don't just turn on and off by themselves. Adjacent to them, they have things called promoter elements. And promoter elements are just additional DNA sequence that for example, these proteins called transcription factors can bind to them, and that can regulate whether the gene is on or off. So if, for example, there was some reason that there was some like relocation of something somewhere else and it segregated the gene from its regulatory chunks.
Right. Then you probably couldn't turn it on or off necessarily.
Well, I don't know if that's like a dominant. I don't know how often that actually occurs.
Okay. Well, that's all right. We're nerding it up over here for a sec.
No, but that's a good question.
But I do want to come back to the original piece, which was wine. You've done some pretty incredible stuff with Zinfandel, as in I think you know this topic.
Oh, Zinfandel. Yeah.
Extremely well. Yeah. Hit me with some Zinfandel stuff. What's going on with this?
All right. So Zinfandel, there is a very enthusiastic group of people who like are really, really into Zinfandel. And they, among other things, these people are interested in, does my Zinfandel clone have particularly interesting characteristics? Just like Pinot Noir and Pinot Gris and Pinot Noir have unique characteristics, even though they're all Pinot. So they've done things like, they've partially funded some of my work in the past, the Zinfandel advocates and producers, and done things, and I don't know if it's the same group, but they've done things like Zinfandel safaris to Croatia to find out like who are the what grapes are the parents of Zinfandel. They have not been discovered, although cousins of Zinfandel have been discovered. But Zinfandel is thought to have come from Croatia and then was brought to Italy by Benedictine monks. And in Croatia, it's...
Sounds about right. Sounds about right.
In Croatia, and I'm going to butcher the pronunciation of this, but Zinfandel is not called Zinfandel. Zinfandel is called Kroljenak Kastelanski and also Pribidrag, I think. Is Pribidrag also? Yeah. And then in Italy, it's called Primitivo. And in the United States, it's called Zinfandel.
Mm-hmm.
And it wasn't always obvious, like, how is it? For a long time, Zinfandel was the most produced grape in California. And there was some, like, curiosity of, like, how it got there. And there's some drama related to that. And I can't remember, there was some guy who was, like, credited with it. And turned out that's not true. And actually, the most likely story is that Zinfandel came to California from New York, actually.
Really?
And there was an architect from my hometown. What was his name? There's an architect from my hometown in Newburgh, New York, who is also an avid botanist. And one of the pieces of evidence that support that Zinfandel came to California from New York was this, like, botanical, like, horticultural journal article that he had written. Andrew Jackson Downing. That's the name of the architect. And so there's a cool kind of story and intrigue regarding the migration and, like, the import of Zinfandel to California. Okay, but here's the thing. So we did this project. We sequenced 16 different Zinfandel clones from around the world. So California, Italy, and Croatia. And we wanted to see, like, do they actually vary in potentially functionally consequential ways, so that the people who have these interesting clones of Zinfandel, they can say, I have this, like, superior clone of Zinfandel. And then if you're planting a Zinfandel vineyard, you can, if there are differences between the clones, make more informed choices about what you plant. So
what would a superior version of Zinfandel be? Like, what makes something superior in that context?
The knee-jerk answer is beauty is in the eye of the beholder. And
that seems like a diplomatic answer.
To some extent, people have different tastes in wine and things like that. But here, I'll give a less obvious answer, which is that I believe part of my grape work is the purpose is to facilitate the creative expression of a winemaker. So whatever their creative objectives are, like, let's say they're involved in planting a vineyard, if you know this clone is more like this, and this clone is more like this, and this clone is more like this, whatever you want, whatever you envision as your end product, you can make planting decisions based on that purpose. But when you do grape research, there is a lot of emphasis on what are called quality-associated metabolites. So things like anthocyanins, which are responsible for the pigment in wine. Like, when you swirl the wine and you look up at it, like, it's red, but is it more brick red? Is it more ruby red?
Sure.
Is it more burgundy? Like, what anthocyanins in the wine, that's what they're doing. Or tannins. What kind of tannin profile do you prefer? Or in whites, I think there are what are called terpenes. So, like, these kind of pretty floral smelling aromas. So, yeah.
Okay, so it really is you weren't joking, it was in the eye of the beholder, and it could be they want a particular color, they want more fruity, they want more earthy, they want more, these various things is what they're sort of considering to be a superior Zinf.
Yeah, winemakers are very thoughtful people. They are generally not like, just like mashing some grapes and letting it rip. Like, they have many of them that I've interacted with, they have a goal of, like, what they're trying to create. And part of that is knowing very well, like, what you can expect from the grapes that you've got to work with.
We chatted a little bit offline, but one of the fun facts that we were kind of looking at is that sommelier exam.
Oh, yeah.
And it turns out something like only 296 people ever have passed that exam. I think that's the right number. I think it's 296 since, like, it was first invented in the 1960s, which means it's literally harder than becoming a SEAL, like, twice over, like a Navy SEAL twice over, which is kind of crazy if you think about it. That's an unbelievable test. That said, you were talking about sequencing things for Zinfandel, among other things, which kind of leads into the startup.
Okay.
So
yeah.
speaking of sequences, you're I don't know what your official title is there. Head of something or other. Head of awesomeness. I'm going to say that. Head of awesomeness.
Yeah.
And you're working with
I was going to try and come up with like a clever title, but I wasn't.
Senior Vice President of, making cool wines.
I'm technically the head of science at Genrait.
Right. And you're working with Taylor and Razib. Razib is also on this podcast, geneticist Razib Khan. And you guys are working on effectively like a genomic-focused data store.
Yes, and there's other cool stuff about it too.
Yeah.
But so doing genomics is not trivial. It's not easy to do on your average laptop. Data sets are really big, and if you're working for a company or an institution that wants to do genomics in an ongoing way, over time, you're generating more and more over time, and you want to be able to make use of all that information. So we make the storage easier, the processing easier. If one of our customers, they have customers and they need to deliver analyses to their own customers, we make that sharing component easier. And one thing we're really excited about doing in the long term is facilitating better contextualization and understanding of your results. And part of that has to do with like the founder. So Taylor, you've met Taylor. Her and the founder's vision is that our company should have some hand in making precision medicine a reality. And getting everybody, I'm sure a lot of people who are listening have had their genome sequenced, and now you have all of this information. Like, what does it actually mean? And as a scientist, okay, I generate sequencing, I did this analytical process, and now I have this result. The next step that is very challenging, it's the most fun part, but is the most challenging is the understanding the story and the data. So that is the long-term vision for the company, not just the those key building block elements, but the stuff that makes scientists excited about being scientists. We'd like to have a hand in doing that.
Right. And again, science being the search for truth, effectively what you're looking for is what would be true for this individual person. Again, sort of a dream state. What would be true for this individual person if we gave them X molecule, in other words, X drug or X medicine, et cetera. And I have to agree. I mean, that would be, I think that would be a dream state for me as well. For most people around the world, personalized medicine would be incredible. We're not there, but it seems like you guys are trying to push us there. That's great.
Yeah.
It's exciting stuff.
And that piece, the bigger vision, and then thinking about, okay, what are we actually going to do to get closer to that thing? That's, among other things, the most, is one of the reasons I joined the company in the first place. Other than you've met Razib and Taylor, and I don't know if you've met Shantanu, but the founders are great, great, smart, competent, able. So I'm just I'm happy to be Ringo, not an original Beatle, but happy to be the
That's
fourth.
fair. I'm going to sidetrack for a half second. Ringo was the only professional musician in the Beatles. You could literally ask anybody about this. I looked all this stuff up. Ringo was the only one who got almost everything done on the first take. I mean, they all eventually became amazing musicians, but Ringo was actually a pro before.
That's cool.
It's actually pretty sweet. So I know Ringo's the overlooked Beatle, but it's no, I mean
he's not overlooked. He's my favorite.
He was a pro. He's my mom's favorite. He's always been okay. Cool. But besides
Ringo, back to science.
Actually, more back to you because I'm curious. It seems like a really amazing vision. Is there something in particular that sort of attracted you? Because you were working at, and I do want to talk about cancer at UNC, but that's what you were kind of doing previous. Is there a reason you wanted to make the move to this particular firm outside of, the cult of personality?
Oh, no, I would. Okay, so as I disclaim, I am proud of the work I have done, and I admire the people I have worked for. I have known for a long time I do not want to be an academic. Okay. And actually, while I was interviewing at UNC, I had been interviewing at companies, for example. I have this, how do I say this? I have a almost like whole, like kind of reverence for entrepreneurs and for commerce that I don't always observe. I don't observe enough in academia. And I also have, whenever I have an existential crisis when I was an academic, I'd be like, oh, I should just like quit my job and be a winemaker. I'd love to do that. And that comes from I love being a scientist, but I kind of crave the satisfaction I envision you get when you can say like, I made this.
Mm-hmm.
Or, I worked with this group that I, adore to make this thing, and I can hold it in my hand and it has real value to other people that is measurable. I crave that thing.
I mean, that's a very real thing. I know there's a lot of bricklayers out there who literally have the exact, people are always like, well, it's just a bricklayer. And it's like, no, a lot of those people are some of the happiest people in the world. They can literally point at the end of almost every day and be like, I built that, I made that, it's real, it's measurable, it's valuable. It's like a perfect way of putting it. I also have some reverence for some academia folks, though. I mean, some pretty amazing stuff has come out of academia. But there's no shame in saying you're a capitalist, too. There's nothing wrong with that.
Yes,
I'm not saying you're doing it for the money. I'm just saying, making something real and getting paid for it and having market value. Nothing wrong with that. That's great. Let's go into the cancer piece at UNC because you did some pretty amazing work there.
Oh, thank you.
I think you were working pretty heavily with melanoma, if I recall correctly.
Yeah.
Let's talk about that.
All right. So I was working with this group who has this, I think the project's been going for like 10 years. And they study the project I was working on was taking stage 2 and 3 melanoma patient samples, and those samples have a very wide prognosis, I think is the right word. Like some of them have better outcomes than others.
Mm-hmm.
And the purpose of this project was to determine whether you could use genomic information to better predict the risk for those kind of ambiguous samples. If you can do that, if methylation information or genomic information has prognostic value, then when you go to your doctor, they can better push you in one direction treatment-wise or not. So you can better say like, this is dangerous, go get treatment, or this is not dangerous, have a nice day.
You said a word there. You know what I'm going to ask? Methylation. This
Okay.
isn't methamphetamine. This
No.
is definitely okay. And so this is something else.
Mm-hmm. So methyl marks or methylation, it's often talked about in terms of epigenetics, so changes to DNA that do not change the sequence.
Which is fine, but I want to give this in the context of what you're just talking about,
Yes.
the cancer context.
Right. So this group that I worked with, so this is where I was going with this. This group that I worked with had previously published a paper about what's called the CIMP phenotype, or the CpG island CpG island hypermethylated phenotype, which means that in some samples at the same stage of melanoma have this like global hypermethylation, and this is riskier. This group has poorer outcomes, and so CpG islands are places in the genome where there's like lots of CGs, and they are methylated. And off the top of my head, in general, it's thought that like methylation, the effect of methylation is that it reduces gene expression. It also has a beneficial effect in that it keeps jumping genes from jumping around. But back to this group, what we did is we measured global methylation in melanoma stage 2 and 3 melanoma samples, and then looked to see and we saw that these CIMP samples, these very globally highly methylated melanoma samples, have poorer outcomes. The consequence of this is that there will be a day when you can go into your dermatologist and when you get your melanoma checked, there will also be an assay attached to that's measuring the methylation level in the sample. And if the methylation level is this kind of intermediate or high level, you're at much higher risks. And so you will make different decisions. That's the value of the project.
Well, that's interesting. Okay, so and the methylation, when we're talking about that, and again, that was perfect context for
cool.
the cancer. But when we're talking about methylation, because I kind of cut you off, I didn't mean to.
No,
I just
sorry.
really wanted to understand the cancer thing. But when we're talking about methylation in general, you said that this methylation or hypermethylation, it reduces genome expression, or sorry, not genome, but gene expression. And then I guess there would be hypomethylation, which is like not enough to where genes can like jump around and do crazy stuff, something of this nature. Is that a fair characterization?
Yeah, so not all hypermethylation is bad. Not all hypomethylation is good. It's context specific. So under certain contexts, you're going to observe a particular pattern of methylation. So a context in which hypermethylation is really good is keeping transposable elements in the genome silent. When they jump around, they can cause disease. But in a negative context, in these melanoma samples, like there's something about that puts the patient in a higher risk group that is more likely to die within 5 years of diagnosis. Methylation is also relevant in terms of, so some other projects that I worked on were to evaluate, does drug treatment affect methylation pattern? And then under certain disease states, you can have changes in methylation during development. So methylation is not necessarily set in stone. It's flexible can be flexible during development. So that's an amazing study. So the question has to be asked, of course, are molecular biologists the brains behind the medicine? We're just a piece. We're a piece.
Okay.
Yeah, there's a lot of hands in making the soup. And crap, what I was going to say. Oh, I forgot. It must not have been important. But it takes chemists, it takes physicians. So most of the people I worked with, they were physicians and researchers.
Which makes sense. It seems like you and chemists would be like joined at the hip.
Yeah.
Almost, right?
Yeah, I love chemists or I love chemistry. And my first mentor was a chemist. I mentioned is a brilliant chemist. You just, you only have one life to live, so pick,
Two things jump into my head when we say chemistry. One of them is that I would love to have someone on like this, but when chemistry started entering sort of the gastronomical world, which I think wasn't that long ago. I think it started in the 60s, but it really ramped up very heavily in the 80s and 90s. There was just sort of this explosion of crazy things you could do. Like no one ever heard of like deconstructed foods and all these like crazy things that people do on the Food Channel and like that like a little bit of foam or whatever. And you're like, wow, why does that taste like a melange of crazy stuff? And chemists really entered that field. And of course, that's an amazing thing. But of course, with a little bit of, pop culture brain, unfortunately, in my head, obviously the next thing that comes up with chemistry, and maybe it's because you said methylation, is obviously Breaking Bad. Like,
yeah. I don't know anything about that.
Well, it was a good show. I'll put it that way.
Yeah,
really liked that show. Regardless, so it seems like you and chemists are kind of joined at the hip when it comes to this stuff. With the cancer research at UNC, like, I know you're not an MD or anything, but do you see a state in the future? Pick a time frame, 5 years, 20 years, 50 years. Do you see a state where we can effectively identify, like, well, I got your whole genome mapped, so I know I can effectively guess what a damaged cell looks like, or with a high probability, and then either eliminate that cell or maybe repair it. I don't know enough about what I'm saying to really.
No, it's okay. I think I know what you're getting at, which is like can we correct disease
effectively?
Yeah.
Right. I mean, cancer in particular is it's your own body. It's damaged cells. Well, if I've got a sample of your regular cells, I mean, I wouldn't mind getting like a whole bunch of needle pulls from various places in my body. If they're like, yeah, okay, we have a whole bunch of cells, all of them are functioning. If you ever get cancer, we can detect it effectively instantaneously because it's a pattern matcher, and then we can like search and destroy those. That would be amazing.
Yeah. So level one, in my brain, level one of like, I have my genome sequenced. I know for which diseases I am at relatively higher risk. And so I can go to my doctor and say, I'd like to check for this maybe more frequently than would otherwise be called for, or maybe I would start getting screened for things younger than I would, because they, you always hear, you catch things early, the better the outcomes, the earlier you catch things. So there's that piece. And then there's the piece of like, well, can I, edit out the problem? And that I think is not too far away. I don't really know the mechanics of how CRISPR works, but that is what people are talking about. And it seems most likely that simple diseases caused by simple mutations, like one change in one gene, that seems like the lowest hanging fruit.
Nice.
But a lot of disease associations, these 100 variations are associated with higher risk for this disease. Though that's not necessarily a causal link. So it's not necessarily true that these variations are all causing this. But even if that were true, that would be more challenging to build.
Okay. But could you see a
It's
path for
doable.
that in like 50 years?
Yeah, anything is possible with the mind.
Okay. All right. Yeah, I mean, I tend to agree. All right. Well, let's go with something slightly happier.
Yeah.
But I do have a little bit of a moral conundrum for you. So you're a molecular biologist.
Mm-hmm.
Would you have a problem? Have you seen Star Trek, any of the Star Treks or anything like that? They've got replicators.
I've watched a little bit of Star Trek, but I'm not like a Trekkie. I would lose badly at Star Trek trivia.
That's fair. So they've got replicators. Replicators effectively, and theoretically this is possible, you would just need several atomic bombs worth of power. But what they basically do is they recombined molecules straight out of the air into like an apple or a big fat tomato or something like that. My question is, that tomato had no genesis. It was literally put together by high tech, and obviously Star Trek is in the 24th or 5th century or whatever. And I mean, this is of course hypothetical, but would you have an issue with that if basically it didn't really come from what we would consider today to be life, but afterwards, I mean, the seeds are intact and like you just have this big giant fat tomato? Would you be all right with that?
First, well, short answer, no, I have no problems with very big fat tomatoes. I hope they're still flavorful, and there are other characteristics besides the size of the tomato that I would care about. But no, I have no problem with a big fat tomato. And I would also say, like, all the other tomatoes are still low tech tomatoes. Like, you're walking through, a forest, you're not going to find juicy tomatoes on the vine that you see at Whole Foods, So I see the big fat juicy tomato of today, really, and tomorrow. Like, this is just like we have better tools to do things that we've already been doing for a really long time.
And that's effectively where I was going, right? Star Trek is sort of the end state where we literally pull molecules out of the air and just create this. But it's more along the lines of like basically coming back to now, which is, okay, you have a tomato, you pull some seeds out, you plant another tomato plant, and so on and so forth. Right, sure. And we would consider that, okay, that's, all organic and everything's good to go. And I guess the question is, what if we genetically modified it so you have this big fat tomato? I mean, is that a bad thing?
No, I don't see a reason. And if anything, there's a lot of reasons to do that include, like, organic farming is difficult, especially if you live in a place that has a lot of pest pressure. Genetic modification means likely more resistance to stress, so like drought, salinity, flood, things like that. That's a good thing because, being able to feed people is a good thing. There was this, for a long time people thought, oh, we're not going to be able to sustain this many people on the earth. It's genetic modification and access to markets that mean that we don't have mass starvation. That's a beautiful thing. Then aside from resistance to stress, that means you could also use less pesticides. So if you're concerned about if you love your like hippie tomato that has not been sprayed with anything, genetic modification increases the likelihood that maybe you use less pesticides or don't have to use them, or you don't have to use as much fertilizer or water. If you're concerned with the preservation of natural resources, genetic modification is one way to reduce impact. If that's what you value, this is a good solution for that.
Mm-hmm. So I think a lot of people, just to be perfectly honest, I think people aren't really concerned with like, yeah, I mean, they're making it somewhat pesticide resistant. So like those are the good side effects, right? But I think a lot of people, they're here molecular biologists and it sounds like you're playing God and whatnot. And they're afraid that you make these other modifications where it's like, and I'm also going to like drop your IQ points and make you subservient. You know what I mean? Like maybe that's all crazy BS, which is fair. But I think a lot of people are concerned with those types of things. How do we assuage those people's concerns? I mean, I don't think you have to, like, I'll give a quick example. I don't think you have to in India when I think it was Norman Borlaug in the 70s came up with dwarf wheat. He won a Nobel Prize. Basically, it doesn't topple over in the sun. It's short, but it produces the same amount of wheat, which was pretty amazing. I mean, that was one of the first like global type of deal, global GMO type of deals. But we kind of backport that to the U.S. and now you've got, whole sections of grocery stores or even just entire grocery stores that it's like, if it's not organic, we're not putting it in here type of deal. And then you have other grocery stores where it's like, no, we really do have, like we've got all the regular stuff that people are used to, but we also have like organic sections and whatnot, which is great. But yeah, I mean, some people are genuinely afraid of like scientists. I don't want to say like yourself because that's rude, but like of scientists kind of playing God and being like, I'm part of this big master plan. And I don't know, you get into these crazy conspiracy theories where.
So let's say you're afraid of, but besides the technology itself, like you're afraid that if we make the big juicy tomato, I'm not going to get the tomatoes that I like that are not the big juicy tomato, whatever. My point of view is that markets tend to give people more choices, not fewer. And there are some, asterisks here, but generally, like when I go, if you just walk outside today, go to a grocery store, there's lots of different kinds of tomatoes, there's lots of different kinds of apples, there's lots of different kinds of all kinds of things. And I don't see why it's necessarily true that, we have this technology that makes this new one thing and you're never going to have any other choices again. And so long as capitalism can exist, there will be, and provided there is a demand for non-big juicy tomatoes, I tend to think that they will persist.
Which is, I mean, personally, I would tend to agree with you. But again, like you definitely have people who are wary of various things when we start tampering with. Well, I think I don't know. Maybe that's a biased word. I'm kind of on your side. I like to tamper with things. I think it's fun and interesting, and what can we do, and so on and so forth. But I mean, I would tend to agree. Usually, you don't see a lot of people where it's like, yeah, less choices in the market is a good thing.
Yeah, yeah. Like that whole thing. Have you ever heard of like choice? It was in the pop culture a while ago of some people get paralyzed by choice or something
analysis
like that.
paralysis. That's a very real thing.
Yeah, but when I see, 20 different varieties of canned soup, and I have guinea pigged myself in tomato soup studies, that's a story for later or another day or whatever. But I am relieved by that. And I do take a really long time in a grocery store, but like part of the reason is just like, what do I want to buy? And part of it is just like basking in the glory of like 20 varieties of soup.
So we're going to talk about tomato soup now.
Oh, yeah.
So what does a molecular biologist do when she's sampling 58 soups or whatever this is with tomatoes? And I think we've kind of spoken about this offline, but like, if I recall correctly, you're more of the sort of acidic and non-big fat creamy type. Yeah,
I do. So I not only enjoy doing science, but like weird thing about me, I also enjoy being a guinea pig. And when I was an undergraduate student, you could sign up for all kinds of studies, and at the end you get like some beer money or whatever. Or now they do Amazon gift cards, but they used to just give you cash, like sit in a room and answer a questionnaire. And so psychological studies, business school studies, and food science related studies. And so at UC Davis in particular, I would do these every once in a while. One of them was a tomato soup study where you don't necessarily know the purpose, like the main research question, but I would just go in at lunchtime and I would sit in a dark room and someone would raise like a little door and slide some tomato soup in front of me, and then I would taste each one and then rank on a sliding scale like how creamy it is, how acidic or salty, how do I taste other things. So going through like how, what are some other things? Is it spicy? Is it creamy? Is it, sweet? Is it sour? All that kind of thing. And those are just fun for me because, I really do enjoy food and smells and just, it's a fun thing. Yeah. With wine when it's done. So these are really fun. And if you're interested in these kinds of things, just find like a university near you that has a food science department. I'm sure they do something like this. I would get together with a group. So the researchers would host a group and everyone would come in around lunchtime and they'd have us smell and taste some wine. And they'd ask, please, tell us what you smell. And then we'd write it all down. And then the next time we met, the researcher would put that thing in a glass. And then everyone in the research group would smell the glass and say, when you said it smelled like dirt, is this what you smelled? So we develop a common vocabulary for doing the study. Yeah. There are some smells that are very similar. I think like maraschino cherry smell and almond paste smell are actually the same.
I think I've experienced that. I think I agree with you on that one. Yeah.
So when you're in a sensory panel, people's like, oh, I smell maraschino cherry, I smell, whatever. They'll put the maraschino cherry in the glass and everyone will smell it and be like, okay, when you smell this, we're going to call it this.
Right. Wine vocabulary is very specific. You have to really, really get into it because a lot of people smell and they're like, well, I smell wine. It's like you've got to get underneath that. A lot of them have some really, really interesting notes. Okay.
And one of the reasons for doing these kinds of things, so I do it for fun. I love being a little guinea pig, but another study was about, California has fires. Vineyards are affected by these fires.
Big time.
Question, what is the effect of the fires on final wine composition and quality? And what then is the effect on the price a consumer is willing to pay for those wines that were made in years where there were these massive fires? So this is kind of like how you get from like smelling and tasting things to the
macroeconomic
world.
Sort of climate type of deal. Okay, that's interesting. That's good stuff.
It's fun.
So when you're doing molecular biology, there is a rumor that you can take these, what do you call it? Glowing stuff? What's the word for glowing? It's
fluorescent.
Fluorescent.
Or bioluminescent.
There it is. So there's a rumor that you can take this bioluminescent stuff and make things glow.
Yes, you can.
You make my hand glow?
Yes, that is doable. Although I don't think it would be permanent. So you would be doing what's called a transient transformation, and it would probably, almost certainly last under a month, maybe a few days. I haven't actually tried it. I know in fruit, in like strawberry, a transient transformation doesn't last that long, like maybe a few days or something like that. I don't know how long it would last in a human, but probably not long.
Here's the thing. When you say not long, I was thinking seconds. When you're telling me a couple of days, I'm like, that's awesome.
That would be my guess. Although, you can fact check me. Is it a few days or like a week? I don't think it would be that long. And that's because your cells have ways of saying, that doesn't belong here, and destroying it.
So in other words, you could make a human glow, or at least parts of a human.
Yes.
I feel like there's a definite business here. Like there's no way that people wouldn't actually want to be like, oh, awesome. Who wouldn't want to show up to like a cool party, like a rave or whatever, and your whole freaking face is literally like lit. And everybody's just staring at you like, holy crap, what did he do?
Okay. There are very serious reasons for using green fluorescent protein or bioluminescent proteins in research, for example. If you attach, for example, green fluorescent protein to some gene, or the gene for GFP, or green fluorescent protein, if you attach that to a gene of interest, then when the gene is expressed and it has this GFP attached to it, you can see where it goes in the cell. Like you can see, under a microscope, you can see like, is this protein associated with a cell wall or a cell membrane or a nucleus, or like what's going on? Is there a lot of it? If you're doing an experiment where you want to control the expression of a gene, or you want to understand how is this gene regulated, you try and turn it on and off, and you can measure, is GFP going up or down? So you can use it as a kind of reporter to better understand gene expression and regulation. But you could also make your hand glow, and that would be fun.
That would be really fun. I mean, I'm going to say it, there's no way there's not a startup here.
Yeah, or like
where you can basically send things that people make certain parts glow or whatever. And then, I'm going to stop it right there. But like various parts glow that they would want it to glow. I'll just say hand.
Yeah, but like,
that would be amazing. Like all of a sudden you're dancing and you're waving and everything like that, and your hand is glowing.
Yeah. Who needs like glow
no,
sticks?
Forget glow sticks. You got 5 of them attached to your hand. That'd be unbelievable.
Yeah. Yeah. I wonder. Or actually, okay. Let me think. So I think there are different bioluminescent proteins, and to be fair, I don't know or remember which ones you can turn off a light and actually see glowing versus you need to visualize the glowing protein under a certain wavelength of light. You know, like when you go, I don't know if you ever do, what's it called?
UV?
Bowling. When you go bowling and they have like if you wear white, you glow.
Yeah.
I don't know.
It's UV.
I don't remember off the top of my head like which kinds. Yeah.
That's really fun. I was expecting UV. You mean you can make something glow in like regular light? That'd be crazy.
I don't recall, Senator. But I think, fireflies light up. There's fish that light up. You don't need special.
That's right.
Equipment to visualize that.
So how does the body expel that stuff? Like let's say you made my hand glow and then 3 days later it doesn't. Is it like sloughing off the skin? Is it like just like
so
I don't know.
I think there's a few layers of like, that doesn't belong here, get it out. One of the ways, although I don't know if this is, one of the ways. So if you were to attempt this, you're inserting foreign DNA into your cells. And there's a variety of ways to get it in. You can use liposomes. You can, I'm not talking about like your hand, just in general, how do you transfect cells? Like in a tube, you can use chemically competent cells. You can electrocute them. And then also you can use liposomes, which kind of go, and they interact with your cell membrane, and then I don't know.
That one sounded better than all the rest of them.
The sound really summarizes it.
That one sounds way better than the rest of them.
Okay. And, but once it's there, you have double-stranded DNA present. So you have your cells have what's called RNAi machinery that recognizes double-stranded DNA and wants to chop it up. And this is also, at least and there's some differences between plants and animals, but at least in plants, RNAi machinery recognizes double-stranded nucleic acid or DNA or RNA. DNA, and then chops it up. And this is a really great way to defend against like viruses, for instance.
Okay.
Then the other question of like, and I hear people talk about this in other, how do I want to go about answering this? There are vesicles within your cells that are the garbage disposals of the cell that take, basically accumulate garbage and then destroy it.
Okay.
So that there's that as well, but off the top of my head, I don't really remember the mechanics.
Oh.
But yeah, there is a kind of there are little garbage collectors in your cells, and there are mechanisms within the cell to recognize stuff that does not belong.
Makes sense.
Mm-hmm, mm-hmm.
The genomic diversification of grapevine clones.
Oh yeah, this is the Zinfandel thing. So
that's a paper you put out.
Yeah.
And so what does that mean? Because when we, and again, I'm not trying to sidetrack. I actually want to go back to it, and specifically because there's no sex going on. Therefore, the plants are cloning themselves.
Or we're cloning them.
Or we're cloning them. Is that what we're doing?
So grapevines, when you plant a vineyard, you're not planting seeds. You're basically planting cuttings. Means there's no sex going on. So, and there's an asterisk, they should be genetically identical. But grapevines have been cultivated for hundreds and hundreds. So Zinfandel has been cultivated for hundreds and hundreds and hundreds of years. And mutations occur, and these can spread. And basically, that's how you get different clones that are, the only way in which they differ is somatic variation. Or, mutations that occurred in like one cell that proliferated through a cell layer, and then someone walked by that vine, like a vineyard manager, and was like, oh, that's cool, and lopped it off, and if it was good, planted it, and now you can propagate this unique clone.
Hence Pinots.
Pinots, right?
You had a. I don't know what was the progenitor of all this, but
yeah, I don't remember the order, but there is a paper on
one
that
way or the other.
thing.
Someone literally looked at either a red or white vine, and there was the opposite color.
Yep.
And went, whoa, I should plant that. So that's really what's going, or for that matter, a gray one or whatever it is. Right. So we're doing this. Okay.
Yes.
Does this happen? Like, what's the prevalence? Like, you're walking up and down, a vineyard, and there's a bunch of Pinot, and it's like, oh, look, there's a bunch of Pinot Gris all planted everywhere. And then you're like, look, Pinot Noir. There's literally a vine of it. Is that, could you just do that? You walk a couple hundred meters and it's just there?
Yes. So vineyard managers will find what are called bud sports that have this reversion to either colorless or colored grapes. But this whole thing of like somatic mutation in clonally propagated populations is like not just cool for grapes. There's this concept called Muller's ratchet. This is like a basic biological principle that in the absence of sex, asexually propagating organisms, or like clonal populations, will accumulate putatively deleterious mutations. And if it's a diploid organism, they'll be heterozygous. So you basically are like loading up. You can load up on mutations over time. So grapevine is a really useful model to study Muller's ratchet specifically because you have clonally propagated grapevines for literally hundreds and hundreds of years. So like part of that paper was trying to understand, just like on the basic biological level, what happens to genomes what happens to genomes of clonally propagated organisms over that kind of time span. And so we looked, how much mutation is happening in a gene, and then within the gene in the exon or the intron. So the exon being the functional part, the intron being the part that's spliced out during transcription, or how much is happening in the intergenic space. So the area in the genome, which is most of the genome, that is not genes. And the finding was that after accounting for differences in the amount of those elements, so like I said, like there's very small amount of genes, gene length, and there's a lot of intergen. So after accounting for that difference in amount, we still saw a lot more somatic mutation happening in intergenic space. And that gets back to our melanoma discussion and methylation discussion because so methylation is a great tool that the cells use to keep transposons from jumping around. Transposons were initially discovered in corn by Barbara McClintock, who was a scientist at Cornell, and she won a Nobel Prize for it in medicine, I think.
Shout out to Babs.
Yeah. But, okay, as a consequence, so you have this intergenic space that has transposable elements in it, and it's very highly methylated. It's kind of a double-edged sword, so there's a trade-off. So methylated cytosines, so you have A's, T's, C's, and G's. The C's are more prone to what's called spontaneously deaminate. So like without impetus, just they'll spontaneously deaminate to uracil, I think, a U, which then, yeah. So you're going to see as a consequence of transposable element methylation, your higher rates of somatic mutation in intergenic space. And this is the cost that the cell pays for methylating transposons.
Which is fair. I'm confused, though. You said a C trans some things to a U?
To a U, yeah.
There's
U's
a U in
RNA.
genes?
In RNA.
Oh, in RNA.
Yeah.
Okay.
Yeah.
Thought you were just adding letters. I was like, cool.
Yeah, I think, no, yeah, so there's the U, and usually you only see it in
Given
RNA.
that we're doing a bunch of different cloning, et cetera, let's just say you had a time machine, you go forward 200 years. Is wine still wine?
Well, places like Cornell, I know, have released a variety of new cultivars. Is wine still wine? You know, okay, the most optimistic take is like the old world will still be doing what the old world does, and the new world is like the wild freaking West and is always kind of experimenting and rolling the dice, and you can find really amazing things, you can find really weird things. And that kind of spirit of innovation in wine, I think, will continue to happen, and there will be new and cool things in the years to come. Okay, the other piece, though, is like things like cold, for instance, things like pest pressure cause a lot of crop loss, and to kind of circumvent this, people breed novel varieties of grapevines, and these might not be genetically modified. They might just be cultivars that are bred using traditional breeding techniques to make grapes that ideally taste like the cultivars that we love, but are also cold resistant or drought resistant or whatever. Yeah, and I know the face.
Isn't that just genetic modification using different methods, or for that matter, slower methods?
Yeah. But there's a variety of ways. There's more than way to. There's more than one way to skin a cat, as they say.
Fair.
And there
That's a brutal saying.
It is awful. So actually, I say that. It's such a handy.
That wasn't a dig at you. I'm just saying it's a brutal.
It's such a handy saying, and I actually, my former boss was Italian, and I said that to him on my first day of work, and he was like, "I don't know that saying.
Why are you skinning cats?
That's a terrible saying." I was like, "Yeah, there's no wrong way to. There's no wrong way to eat an Oreo or a Reese's. That doesn't work. But there's more than one way to skin a cat. Like, is there a better one? Is there something similar, analogous? No.
Not that I can. I mean, not that I can think of off the top of my head.
So it stays.
But it's just a brutal saying.
It is. Yeah, like who's the first guy to teach the various ways to skin cats?
I honestly think one of the most irritating ones that I hear is you made your bed, now sleep in it. I don't. Again, unless you're talking to a carpenter who actually made the bed, most people would be like, "Wait, I did the work to make my bed, and now I get to sleep in it." This is the opposite of consequences. It should be, you slept in your bed, now make it.
Bed.
Right.
Yeah.
I just never understood that. Unless people, maybe I'm taking it wrong. People are like, no, it's from the point of view of a furniture maker, a carpenter. I'm like, oh, okay, cool. That makes more sense. Like if you made a crappy bed, but anyway.
I'm sure there are linguists who really know like the history of all of these really bizarre sayings.
We still say cart before the horse. That one's got to be pretty old.
Yeah, but that one makes sense. Like you know, the horse pulls the cart.
Yeah,
you
but
can't put
I don't
the cart
know
before
anybody.
The horse.
I don't know anybody who was like, yeah, that's a good idea. I'll just stick the cart in front of the horse.
Mm-hmm.
What?
My grandmother. My grandmother was an Italian immigrant, and she had a lot of really cool like rhyming sayings. And one of them roughly translates to, like, don't count your chickens, but it's the water is boiling, but the pig is still in the mountain.
I like it. I bet it sounds amazing in Italian, but I'm completely lost.
Okay.
Good enough, though.
Yeah.
All right. So, let's just say for sake, because we already took a time machine to go see if wine was wine. So we're still in the future, let's just say. We're 200 years in the future. What does molecular biology look like now? Are we not molecular? Are we literally doing like cell engineering?
Well, the molecular stuff is smaller than the scale of the cell. So
Really?
like the cell contains the nucleus, contains the genetic material. So molecular is still a smaller scale. What are we doing?
Yeah, what's going on 200 years in the future inside of molecular biology? Are you literally like engineering things from like the quark level, or like are you all basically physicists now? Because there's
Yeah. There's a great webcomic where there's a lot of webcomics of mathematicians, physicists, chemists, biologists, psychologists, No, I think there's a lot of. You'll always need biologists. You always need chemists. You always need the people in the different disciplines. I think the purpose of all of those disciplines is not, at least my point of view, is not generating new knowledge just for the sake of new knowledge, but the purpose is always, the motor is always the improvement of human life and giving people more choice and options and things like that. And so there are a lot of cool molecular biological tools that are being used today. I would like those to be used towards that end and be readily available to people.
So again, we're 200 years in the future. I imagine we have a pretty amazing understanding of what it is to be a human being, better than we do now. I mean, right now we're just like, okay, we're big giant meat sacks filled with water. There's some chemicals going back and forth. There's some very weak electricity going back and forth to various places. And really we don't have too much better of an understanding than that at present. Do you think that this field ends up getting to like, here's what life is?
No.
Like to an almost definitive answer? No.
Here's what life is. Do you mean in the absolute, like, this is the recipe kind of deal, or in like the more like philosophical point of view?
Well, it's not necessarily. I mean, I guess you could say either one of them, but I wasn't really going for philosophical because it's not a particularly philosophical field. Not that you don't eventually get there, especially when someone asks goofy questions like this. I mean, do we end up creating the soup again? And you're like, hey, look, like this is what happened the first time that life was created. Like there's some amino acids, there's a lightning strike.
Like
we literally Dr. Frankenstein it up. Like, what does that look like?
Yeah, so there was this famous experiment called the Miller-Urey experiment where that was basically accomplished. And do you know the Simpsons episode? Were you a Simpsons fan?
Yeah.
There's I think it's a Treehouse of Horrors where Lisa Simpson does a science project and like she makes a life. She makes like a little tiny civilization in a
Don't they eventually
bowl.
evolve and like start using like rocket ships? I think I remember.
Yeah,
that
they attack Bart and then they miniaturize her, and they're like,
That's right.
They refer, they consider her, their god and also defender against Bart. I don't know. Who knows? I think life is. There's always more to learn. Like the acquisition of knowledge is, I see it as like, you're like wiping away sand in the desert and like finding cool pieces of things. There's an entire discipline called synthetic biology where you can, the goal is to take the genome of one organism, put it in another, and transform that other thing into the first thing.
So you could make a pig into a dog.
Starting from the yes, so
I'm
that's
going
like
macro
conceptually,
here.
yeah. So like macro, conceptually, yeah. Can you can take the basic can you take the basic architecture of a cell,
Yes.
for instance, and put genetic material in that cell in a way that fundamentally transforms the end point for that organism? So that's cool. And then, and people have cloned sheep, you know.
I mean, the word for that, or the phrase for that, I should say, would be what? Cellular alchemy? I mean, that's, I mean, in physics, right? That's what, I mean, I think Newton ended up going crazy from trying to turn lead into gold because of lead poisoning. But
I don't.
Is that a fair comparison?
Maybe a little bit. I mean, I guess in my brain, I don't, well, let me think. You can imagine wanting to make humans from scratch, like if you have trouble making humans the old-fashioned way.
You said yes to the big giant tomato, so
Yeah,
I don't mean
so
to
why
cut you
not?
off, but like, would you be okay with literally like, we started off with one individual cell and effectively created a human?
Yeah.
You'd
Yeah,
be
sounds cool.
You'd be good with that.
Yeah.
Okay.
Yeah. I think, you know, a lot of the fear, like, take away the fear from playing God
Yes.
aspect. I do not have that fear. There is the concern of like, what if this is used for evil purposes kind of deal. And
I mean, we are human. Usually when we discover something, we do
we'll
something
do something funky
terrible.
with it. Yeah. Questionable. And to me, like, if that's your problem, you know, there's always going to be new technology, and if your issue are like moral concerns, those concerns exist separate from whatever the new technology is, and you're maybe should focus on that. Like the philosophy, what are the ideas and philosophy and values of the cultural soup in which you're in, and thinking about those, because those exist, separate from the new thing of the day. There's always that risk of, could this be used for nefarious purposes? But like, it's not that there's something particularly weird about this particular technology. People said this about all kinds of technologies as they came up.
I mean,
So,
AI is the big one now. People sign this like big giant thing, like we have to control this. And I'm like, you were literally effectively saying, okay, advanced math is scary.
Yeah.
And it's like, but there's nothing but like, there isn't consciousness. We're not even remotely close to that, if ever.
Mm-hmm.
And you're saying big math could be used for bad stuff.
Yeah,
and it's like, I mean, E = MC² was used for some pretty crazy things.
Sure. But I think the preoccupation for those that are concerned with the, you know, values and virtues and philosophy and things, I think the focus needs to be on those things in the culture and not trying to play whack-a-mole with new technology. And if you can have confidence in the first thing, you'll have less fear about the second thing.
Do you think this comes from a place where people are just afraid because there's too much to learn? I mean, we had Renaissance people 500 years ago. Like Da Vinci knew almost everything there was to know. Obviously not everything, but like the man really, really had like, there wasn't a whole lot of fields where he wasn't, you know, knee deep in at least, right? An artist, mathematician, inventor. I mean, you know, mechanical engineering. He was incredible.
True genius.
True, you know, unadulterated genius. But we haven't had that for a long time. And it's because there's, I mean, you could spend your entire life and you could never know everything about molecular biology. And that is such a specific field. It's like, there's no way. Computer science is another one. I mean, you could probably never know everything there is to know about tomatoes.
Sure.
If we just go back to the big fat tomato, you could probably spend your whole life and you could be like, I can come pretty close to knowing everything about the tomato, but I really just don't know anything else about the world. Do you think a lot of this comes from sort of like people freaking out, as in like we're moving too fast?
First, I don't think you need omnipotence to make measurable, obvious improvements in the quality of people's life and the ability to like self-determine in all the ways. You don't need omnipotence. And proof of that is like, look at all of this. Like there are a lot of diseases I'm not worried about dying from. Yeah.
The most common thing for surgery, I believe, in the 60s was an ulcer, and now you take a pill. Like that is, say what you will about the medical industry and and pharma, but they have really done some amazing things. Some questionable things from time to time, but like again, in every field, you're always going to get some crazy stuff.
Yeah. I'll just like take a leaf out of Steven Pinker's book, which is, and this is similar, do we have personalized medicine? No. But I'm very glad to live at the time that I'm living, and that the civilization in which we live was constructed given imperfect knowledge, but it's real. Like we do know real thing. We are capable of knowing, however piecemeal it may be, reality, and that's really important. And yeah.
What's reality to you?
What do you mean? So hold on, you don't get to just ask that question without more detail.
Oh yeah, that's the big giant one. So what does this look like for you?
It's the same as your reality. We live in a common reality.
Are you sure?
Yes.
Why do you think that? Because we're bumping up against each other in this soup of molecules?
No. Okay. But it just is. I'm sorry, this is like a terrible answer. How do I know? Why should you be confident that when you walk out your door in the morning that there isn't like some magical hole that plunges you into hell?
A reasonable question. I don't know. I'm pretty confident that that's not going to be the case, though.
I'm 100% certain that is the case. And you can't live your life with the kind of ambiguity of reality not being real. Like there are things we take for granted, and built into those things are, well, we're taking things granted about reality such that we can live our lives and make decisions and things like that.
That's true, but you said 100% confident.
Yeah.
I am not 100% confident that wouldn't happen one day, but only I'm 99.9 maybe repeating.
Okay.
But the reason I'm not 100% is because I have my patterns broken continuously, literally because of conversations like this. I have very strong pattern recognition. I'm an engineer. I look at things like X and Y and Z. And a lot of my friends, they're not engineers. Like logic is not the first thing that they reach towards. That's not a diss, by the way. A lot of times logic is a failure because the things that they're talking about are either on a spiritual level or they're on a emotional level or something of this nature. And it's like logic fails at those levels. It's the wrong tool for the wrong job. And yet that's the one that that's what I would say is my strongest tool. And that said, you're okay. And that said, that's why I bring this up of that's why I'm not 100% sure. So, as a molecular biologist, you're literally looking at like origin of life type of stuff. Like you're all the way down there, way further than I've ever been.
In order to do anything, in order to do anything, reality must be objective.
Not subjective.
It must be. And our realities must be identical. I think it's just like a requirement of logic in a way.
True.
Like,
and maybe that is
in order to walk out your door in the morning, there are certain things must be true.
Gravity. There's just a given, right? But my thing is, and again, this is kind of where I go with this is, I agree in order for us to live, and by the way, forget in order for us to live. What about just, it is objectively true that is true, but the human experience makes it subjective. Does that make sense? Like something may be objectively true for one person, and actually for all people, and yet their experiences tell them that it's subjective from time to time. Does that make sense?
Yeah, I guess, but that's still not like your subjective experience is not creating reality.
Some people would argue with that.
You have a...
I wouldn't, but some people would.
Your mind does not create reality, it perceives it. And yeah, you have different life experiences than me. We're different in many ways. But we're still you probably like different movies than me.
I'm sure we
Just
could find at least
because-
one movie that I don't like that you do and one that,
Mhm.
yeah, and vice versa. I'm sure of it.
But that still, you still can't assert that given I have a different life experience, that actually air is made of bubblegum. Like, there, you know.
I agree thoroughly. What I'm saying to you is this is coming from a computer scientist and a molecular biologist scientist. Our views are, again, I mean, and again, like this is just because I've had so many of these conversations. Our views, in my opinion, are correct, but that's just an opinion. I can't objectively say that they're true for everybody.
Opinions are like something else that everyone has.
That's for sure. That's for absolute sure.
That everyone has,
And they-
objectively speaking.
And they all stink. It's completely true. If you were given a magic wand
All right.
and the magic wand can perform one magic spell, and the magic spell is you are allowed to create anything related to your field, what would you create? What would that look like?
All right. I've already said that omnipotence is not required to act. Human action doesn't require, absolute knowledge. But, one thing that frustrates me as doing genomics is that, let's say you do some study. What's the effect of this thing on development? What's the effect of disease on this process or whatever? And you get a list of things with a statistic attached. It is, how do you contextualize that result in a way that's, the current way you contextualize those results is very inefficient. So like I can start, okay, this gene is interesting given my result, and I look through the literature. But eventually, like, you have to finish, you can't just do that forever. You have to publish or like create a product or whatever. It's inefficient to read and synthesize the entire digitized body of like the entire digital record of like man's pursuit of knowledge. It's like just not possible to read through all of it, and then on top of that synthesize a story like about your result, like in a comprehensive way. Does that make sense?
I think what you're saying is, given that everything alive has DNA and you just did your test on X, let's say it's a grape, how is that applicable to humans? How is it applicable to tigers? How is it applicable to anything else that's alive? And there's really no great way of sort of crossing those bounds and making your results.
I'm more getting at like
Relative
the
to
idea
everything.
of like knowledge loss and the failure to maybe see the big picture or like interesting, non-obvious things in your results, because it's just technically impossible to read it all, process it all, and synthesize and integrate all of it. So if you could, we have, like I use Google Scholar, I like read papers, something. Like if I could actually have like kind of an automated extension of myself to take my results and then contextualize them within man's entire digitized body of knowledge,
Yes.
that would be a really big deal. We tend to, in our results, you have the 1,000 interesting things. It's really easy to readily recognize the things that you already know about and recognize. So you're like, oh yeah, I studied this and I see that this biosynthetic pathway is affected, and I can write about that and publish the paper and done. And then you publish the data set and maybe it's never used again. And now you've wasted a lot of money to generate a data set. You haven't gotten the most out of the data. You have probably missed a really interesting narrative or like story in the data. So I would really love to have that, the kind of insight, greater insight into genomics data. That would be massive. I think that would be a transformative thing, actually, to actually make use of the entire digitized body of knowledge is how I'll put it. Right now, on any given day, we can only make use of a subset. So we're looking at this tree and we're missing the forest.
Makes sense. Seems like something you guys are actually actively working towards at Genrait, though. So you're kind of creating the thing that you really want to exist in the first place, which is amazing.
Yes.
Awesome. Amanda, this has been awesome. I learned a lot.
Oh, thank you.
And I'm gonna have to Google a lot.
It was nice to hang.
Even though some of those words I probably can't spell, but still, so awesome. So, speaking of that. Do you want to talk about, I don't know, socials or maybe Genrait's website? Like what's something you want people to visit so that they can kind of catch up with you?
I am not a very social person.
That's fair.
But you can go check out the Genrait website, GEN as in Nancy, RAIT as in Tom.com. And then the founders are really cool, and I know they're out there. So Razib Khan, Taylor Capito, and Shantanu Das.
Awesome.
Check them out.
That's great stuff. This has been amazing. Thank you so much for coming on.
You are very welcome.
And we're definitely going to go get some burgers after this.
Yes, excellent.
All right. Thanks. For those listening, and on behalf of Amanda and myself, we have been standing on the shoulders of giants. Thank you.